Okay. Good evening, everybody. I hope I'm audible to all of you. Welcome to the sixth week. Just give me a moment. Yeah. Welcome to the sixth week of this 12-week course on genome editing and engineering. So we are always almost halfway through the course. And this course is taken by Professor Utpal Bora of IIT Guwahati. I'm Sanket Mukherjee, a Prime Minister's Research Fellow from the School of Biological Sciences, IIT Delhi. So, a brief idea about today's this week's lecture is about TALEs or transcription, activator, like, effector, molecules. So these are TALEs, right? And we'll discuss a little bit about its history, how it was discovered, and the delivery methods, which actually remain the same as we have already discussed about zinc finger nucleases. So essentially, what is important to know is that similar to zinc finger nucleases, which were also, was discovered from a transcription factor of Xenopus laevis. Here also, transcription is one of the factors, so which is involved. So what is important for us to understand here is that processes like replication, transcription, transcription, these two processes most importantly are involved in interaction with uh, the DNA. So hence, these kind of proteins would be important for gene editing as well. So if we talk about the history, so there in case of talents or tails, there are different RVD domains. So RVDs, these are basically amino acids that show different base pair preferences, and consecutive RVDs in a tail correspond directly to a DNA sequence in the binding site, resulting in a simple one repeat one base pair code. The basic building block of a tail protein is a helical hairpin, and tail proteins contain a series of alpha helical hairpin domains, right? Similarly, in the year 2009, the role of tail tail proteins were identified by a pathogenic bacteria which used to infect plants, right? We'll discuss about it. So this effector protein was type 3 S, basically type 3S secretory proteins. So if you see here, so T3S receptors, they were, sorry, T3S effectors, they have this DNA binding protein, DNA binding role. And so essentially what you can see in the figure on the right, if a normal T2S effector is used, in that case, there is immune response from the plant or from the host. However, if they use the T3S secretory system, which are um, TAL effectors basically, so then there is exchange of genetic material and it is not involved in immune signaling. There is no immune evasion there. So this is one of the important things which was found out. So there are different bacterial secretory systems which are known. So like T1SS or secretory system, T3SS, T4 like this. And you can see all of them in this slide, right? Three of these T3 secretory system, T4 and T6 can also transport proteins apart from uh, uh, other things. Okay. Uh, Okay, so essentially type 3, 4, and 6, they can additionally they can get into another membrane. That's exactly what is written here. While the other ones, they can just get into the periplasm, basically. Now, if you see the xanthomonas secretion system, there are all the secretory systems which are present in a xanthomonas species, all type 1 to type 6. So all these different systems have different functions. For example, type 1 system is for adhesion to host cell or extracellular matrix. Type 2 secretory system, it is used for host cell wall degradation. Type 3 system is used for host cell manipulation and phytopathogenesis. Type 4 system is for bacterial competition and conjugation. Type 5 is for adhesion to host cell. Type 6 is microbial competition. Now, the T3S system spans both bacterial membrane and is extended by a hollow conduit. The HRP pilus, as you can see, here, that probably transverses the plant cell wall. The bacterial effector proteins are secreted through T3S system and translocated in plant cell cytoplasm via bacterial translocon complex that inserts into the plasma membrane. Now, if we look at the structure, if you remember the structure of zinc finger nucleases, it had cysteine and histidine. These two residues were present across zinc in general, but in case of tails, what happens is the C terminal, it contains a nuclear localization signal, as you can already see. So always a protein is written from N to C disorientation. So it has a nuclear localization signal. Downstream, there is an activation domain, AD, and there are 34 amino acid repeats which bind on one base pair of DNA. So all these 18 34 amino acid, each 34 amino acid repeat. So there are 18 of such repeats. So each repeat binds to one base pair. And the specificity of binding is determined by the 12th and 13th base. So these two bases, whatever is present, that will determine the specificity of binding. So if it is NN, then it binds to G, HD, then it's C. So this is how it specificity is ensured. Repeat types have specificity for more than one number of one DNA base pair. Now, tail, when it binds to DNA, so it is observed that it first searches, in the DNA where it can bind to, and then it recognizes. So essentially, using this property where the 12th and 13th residue of the repeat variable domain, we can essentially engineer specific tails which will bind to specific nucleotide sequences of our interest. And if we introduced a nuclease, for example, a FOC1 nuclease, as we have already studied about, so we can also engineer a restriction enzyme as such. Again, as we have already discussed about zinc finger nucleases, the delivery method for TALENs or tails are similar to what we had for zinc finger nucleases. So there could be physical methods like biolistics, microinjections, and electroporation. There could be bacterial-based delivery, viral-based delivery, and chemical methods as well, like liposomes, PEG, and everything. So based on your aim of the experiment, you will design how you would like to deliver. The different tail nucleases are based on fusion of tail with either FOC1 or PVU2 or some meganucleases. So they are like TEV, truncated version of meganucleases. This is one of the like other kind of nuclease. So there are different nucleases which are fused with the tail proteins, and we create TALENs, which is transcription activation like effector nucleases, right? So similarly, we have nicases. So nicases basically what it does is it causes nicks in the DNA. So these are different methods by which we can use. So transcription activator like effectors are produced by what? Virus, archaea, bacteria, or fungi? This is the interactive part of the session. I would expect some answers from you. So if you would like to answer, please. Sorry?
Bacteria.
Right, you're right. Uh, Tail proteins contain a series of repeat variable dye residues, TAL effector, DNA reading, or alpha helical hairpin domains. What would be the answer? Okay, repeat variable domains would be, or dry residues would be the answers. So, I'm sorry, this is not the answer. Alpha helical hairpins are the structural basis of TAL proteins. I'm really sorry for this mistake. So alpha helix are domains. RBDs are sequences that that determine the specificity of tails binding to each DNA base. So that is how it is. Tails are virulence factors of plant pathogens like xanthomonas that are translocated via dash type uh, secretory system in a plant cell. So is it type 3, type 1, type 2, or type 4? Any answers?
Type 4, sir.
Type 3, actually, I think. So if you remember type 3 secretory system that is used for that has TAL-like proteins which are used to secrete or translocate system. Just give me. Which of the following is not a delivery method for talent into living cell? Is it physical methods, bacterial-based delivery, plant-based delivery, or chemical methods?
Plant-based delivery.
Yeah, that's not there very clearly. So we have already talked about this in the slides. So all the other three methods are present. However, plant-based delivery is not used. Talent can be deployed in the form of RNA, DNA, or protein using various delivery methods into living cells, such as physical methods, like we have biolistics, transformation, microinjection, or electroporation, bacterial-based delivery, viral-based delivery, or chemical methods that utilize transfection reagents like liposomes or PEG. Okay, so this is the answer. Full form of STAR is sequence tail assembly reaction, simple tail assembly reaction, simple talon assembly reaction, or simple TAL assembly reaction.
B.
You are right. Simple tail assembly reaction. So STAR, it addresses the shortcomings of existing golden gate or solid phase assembly protocols, which use tail transcription factors for diverse applications in mammalian stem cell and synthetic biology. So STAR uses an isothermal assembly, also known as Gibson assembly. It's labor and cost effective, accessible, rapid, and scalable. So here a small 68 part fragment library is employed and specific tail repeat sequence is generated within less than almost approximately 8 hours. So uh, this is about TAL. Choose the correct sequence of tail assembly methods. Is it 2000? I mean, you can see the options. You can tell what's the right one. So option D, so this is how it actually evolved. 2010, in 2009-10, we the role of tail was discovered, after which 2011 we got details. In 12, we got techniques like Flash and ICA, and in 13, we had the do it yourself protocol. In 2016, there was discovery of STAR, and in 2021, it was DCTA. So option D would be the answer here. Lauren Tesson and colleagues first successfully created gene knockout rat using customized TAL effectors in which year? Any answers? So the year is 2011. This is the study in Nature Biotech. It was published. HIV-1 infection of CD4 T cells involves binding of the viral protein, glycoprotein 120, to the primary cellular receptor dash and either of the co-receptors dash. If you remember, we have discussed about it in the last slide also. CD4 is the primary receptor and CCR5 or CXCR4. So option B would be the answer. Actually, yeah, so option D will be the answer. CD4 and CCR5 or CXCR4. These are the co-receptors which are used for the entry of HIV into the T cell. A mitochondrial targeting signal, MTS, is fused with TALEN to allow localization inside the mitochondria. Um, well, and which part of TALEN is it fused to? Like N terminus, T terminus, C, or A? Any answers?
N term.
Uh, you are right. So as we said, N to C, we have this orientation. So in the N terminus. Full form of crab in tail crab is knockdown associated box, Kruppel associated box, Kruppel assembly base, or knockout regulatory associated box. Tail crab. So it's Kruppel associated box. You can read about it in your lecture slides. What is the basic building block of tail protein? Is it beta sheets, helical hairpin, alpha helices, or beta loops?
B.
Yes, helical hairpin. Again, the same question coming up. Which of the following is not a physical method of delivery of talent? Not a physical method. Is it biologistic transformation, microinjection, electroporation, or transfection reagents?
Electroporation.
Electroporation is also a physical method. So, basically, transfection reagents like we have lipofectamine, liposome mediated delivery. Those are not physical methods. So that those are chemical methods. The rest three, biologistics, microinjection, electroporation, all three are physical methods. Which when was do-it-yourself protocol introduced? Was it 11, 12, 2011, 12, 13, or 16?
2013.
You're right. Which of the following is an advantage of gypsum assembly? There is need for specific restriction sites. There is no need for specific restriction site. Joining of fragments is specific to the sequence. Or is it you can combine only one DNA fragment at once in a one tube reaction? There's no need for specific restriction sites in gypsum assembly. It's about assembling tails. You can read about it. STAR uses an isothermal assembly called DASH that is labor and cost effective, accessible, rapid, and scalable. Is it gypsum assembly? Is it iterative capped assembly? Is it flash or detail assembly?
A.
You are right. Which animals are model system for investigating human diseases such as cancer? Is it monkey, zebrafish, cats, or elephants?
Zebra.
Yeah, zebrafish is the answer. Talents can be deployed in form of RNA, DNA, or protein using various delivery methods into living cells, such as physical methods. Then we have bacterial-based delivery, viral-based delivery, chemical methods. Okay. So which of the following is correct? Is it only 1 and 3, 2 and 4, 1, 3 and 4, or all of the above? Should be all of the above. When did Lauren Tesson and her colleagues successfully create the first gene knockout rat customized TAL effectors? 2010, 11, 13, or 15?
2011, sir.
11.
You're right. Full form of star, we have already discussed this. Again, it's a repetition. Which of the following is necessary for mitotalin to perform gene editing in mitochondrial DNA? A mitochondrial targeting signal is delivered as donor template for HR. A mitochondrial targeting signal is fused to the end terminal of talent. A microinjection of the talent machinery into cytoplasm without MTS, or all of the above.
B.
You are right. You're right. So we have already again discussed about it. So that would be end of today's session. Thank you all for joining. If there are any questions, you can definitely ask at this time. Any questions? We have very less people now. Let me stop sharing. Anything that you would like to tell?